Okay, so let's go to DrugBank and open this. And then today we are not going to look into drugs. We are going to select targets. And the speciality is even if you type the name of the drug, it will give you the results, and those results are going to be the targets for those drugs. Can anyone let me know what a target might be in the chat? It's just six of us, so it doesn't matter if we're telling something wrong because we are learning. Can anyone let me know? Yeah, so it's kind of a biological molecule. No, it can be a cell or a tissue on a larger scale, but to be very specific, it's a biological molecule like a protein or any other chemical compound. So let me just type metformin, and I have got this as you know the result. It is not for metformin. We have got something, and we'll just try to look into this. And if you click on that, you have got the full name of the drug, sorry, full name of the target, and it says multi-drug and toxin extrusion protein 2. And what kind of a molecule the target is? It's a protein. As I said, targets are biological molecules. It can be even genes, or it can be even like it can be a DNA or an RNA, something like that. And it has few synonyms for the same protein. After that, you have the gene name through which this protein is formed. And then again, you have a unique ID for the protein from the Uniprot KB database. And it says the target protein, the origin is going to be humans, and you also have humans taxonomic ID from NCBI, that is 9606. And coming back to this amino acid sequence, you have the first line, which is nothing but it is called as annotation. Again, name of the target protein, and then you have an ID, and then you have something called as LCL written. So your homework is to search what this LCL is trying to say. And below that, you have single alphabet representation of amino acids. Everyone is aware of that, right? M is methionine. Yeah. So you have single alphabet representation for amino acids. Then you have how many residues are present in this protein, and then the molecular weight of the protein. And then again, what is this theoretical PI? I think this is something you will learn even in biochemistry. Anyone completed biochemistry? No. Yeah, MSI people, let me know what is this theoretical PI. Yes, go ahead, Sumditi. Very good, very good. Nice. So, whatever she said, that is correct. So, you guys know that amino acids, they have two terminals. One is amino terminal and the other one is carboxy terminal. So, there are two terminals. What happens is when pH changes, amino acids might become positively charged or they might also become negatively charged. Are you guys able to understand? So, there is a pH at which it is specific to amino acids and a pH at which the amino acid remains in the zwitterion form. Zwitterion is when there is equal positive and negative charge so that the total charge of the amino acid, it is zero. And I think this is something you will learn in biochemistry class. BTech people, I think you have it in your next semester. I mean, the coming semester that we're going to enter. And also you have the function of this protein, the target protein that has been mentioned here. What are the components in which this protein is present and the functions and the processes that it affects. And you also have the general function written in a paragraph form. And then you have written the specific function of this protein is antiport activity. Have you guys heard of uniport, symport, and antiport? Okay, so again, I think I can give this as homework to you guys. This is something you will learn in cell biology, and also I think um, the thing is, while learning basic subjects of biotechnology, most of the concepts repeat in multiple courses. So you have something called as uniport, and you have something called as symport, and then you also have something called as antiport. Please look into what these three kind of transports refer to. It's basically uh, different types of transports where molecules get transported from outer region of the cell to the inside of the cell. And then you have the domain specific to the function, and then whatever data is not available, you just don't have it. So it is simple. And again, the region of the transmembrane where this protein is present, that is written here. And even I don't know how to interpret this uh, exactly. So as far as I thought, it is referring to positions. There are multiple proteins. I mean, there are multiple these multi-drug toxin extrusion proteins, and they are present at particular locations. And the location of the proteins has been mentioned. That's what I understood, but I'm not very sure on this part. And again, you have the location of the protein here, and it says cell membrane. And for this protein sequence, you have got the gene sequence as well. If you translate this gene sequence, you're going to get the above protein sequence. And you also have the chromosomal location, and you have the locus. And any one of you completed genetics course? No. So in genetics, there is a concept called as chromosomal nomenclature. So what happens is, I think I don't remember all the things, but I can just give you the basic understanding. So this is what we learned in genetics. Okay, so I'll try to draw a chromosome, and so if this is a chromosome, you have this above arm which is shorter. This is called as P arm if I'm not wrong, and then the longer arm is called as Q arm. Are you guys able to understand? And if we come back to this locus, you can see it says 17, that is the chromosomal number, and then it says P, and then it says 11.2. So, as far as I remember, and I also completed genetics long back, so that gene, it is present in the P region, or in the shorter arm of the chromosome, and then why it is 11.2, that is something even I have to recall. I'm not very sure. Then you again have external identifiers. As I have always mentioned in bioinformatics databases, cross-referencing is something uh, that is done a lot, and it also like increases the credibility. It is like you have you're integrating the information from multiple databases, and then you also have general references, basically the papers that support this protein, either existence or any function of the protein, something like that. And you also have the links here, which are clickable. You can go into these links and you can try reading the papers. Did you guys understand whatever I explained so far? Okay. So, and now I'm going to teach you a little bit theory, and I hope it will not be boring. So, if we go for, let's just go for COX-2. Anyone remembers what COX-2 target was for? Search COX in the targets, and let us select this prostaglandin G or H synthase 2. And we have got this. COX-2 is one of the synonyms for prostaglandin G or H synthase 2. Everyone is on this page? And you can see this is not from humans. Can anyone let me know what the organism is? What is the common name of the organism? It says Canis lupus familiaris. Anyone knows the common name for the organism? Canis familiaris? Dog, correct. So, okay, so the topic that I wanted to tell you guys is something called as action of the drug. So, drug and target, they might interact in different ways. And there are different types of targets. For example, there might be enzymes which are, for example, there might be enzyme which is producing a lot of glucose, which is supporting the production of glucose in the body. If we are wanting to reduce the production of glucose, we are going to target that enzyme. Once we harm the activity of the enzyme, the production of glucose is going to eventually stop, or it is going to decrease. So that is how the enzyme is going to become the target there. And there might also be receptors. For example, painkillers. Painkillers are the best example. So in painkillers, what happens is, um, let me just go back to here. Okay, if this is the cell, and if I draw a receptor, and this receptor, let us say that the red color receptor is for pain sensing. For example, if a molecule comes and binds to this receptor, pain is going to be sensed by the cell and it's going to get transmitted or transferred further. Are you guys able to understand? Or you can just uh, consider this is one of the neurons. Although neurons structure is going to be different, it's not going to be like a rectangle. So in this case, the receptor is going to be the target. If we just block the receptor, the transmission of the pain signals is going to stop. So there is not going to be any pain signal transmission, so that the person, whoever is taking the drug, will not feel any pain. Are you guys able to understand? Hello? Okay, okay. So what I was trying to say was, if this is a cell, if black color box is a cell, and let's consider that this is a neuron, and we have a red receptor which is for pain sensing. So if a neurotransmitter comes and binds to this red receptor, pain is going to be sensed by the neuron and it gets transmitted further to the brain. So once it reaches the brain, like pain will be sensed. Unless it's, or we also have one more type of nervous system, right? Even before reaching the brain, we react. The system of the brain which is controlling the reflex pathways. So here what's going to happen is neurotransmitter is going to bind to the receptor and pain is going to be transmitted. If you just block this receptor with some other molecule so that the blue color actual neurotransmitter cannot bind to the receptor itself. So in this case, there will not be any pain transmission. So in this particular example, this red receptor is going to become our target, and whatever we are going to design is going to become the drug. Are you guys able to understand? Okay. And then we might also have, if we consider this as a cell, we might also have some ionic channels, especially we have calcium channels, there are sodium channels, potassium channels. So those things play very important role in few biological processes. If if like transmission of a particular ion through the ion channel is harming our body, our target should be either to semi block or completely block the ion channels, such that so such that there is not going to be any further ion transmission. So in that case, ion channels are going to become the target. And again, we have something called as transporter proteins. Have you guys heard of that? For example, hemoglobin, which is a protein, but it transports oxygen. Hemoglobin is just one of the examples where we are it is a very common example where oxygen gets transported. There are multiple other proteins in the body which act as transporter proteins, and if there is a particular protein in biology, you have something called as similar molecules. For example, if let's say this was a transport protein A and it had to transport a blue molecule, but there is one more molecule which is similar to the blue molecule, but it is not blue molecule, it's a red molecule. Because of similar size or similar shape, this transporter protein fails to recognize the specificity of the molecule, and it will just start transporting this red molecule instead of the blue. In this case, the transporter protein becomes our target. Are you guys able to understand? These are like some random examples that I'm trying to give you to make you understand like what are the possible different targets. Okay, am I audible now? Okay, so what I was trying to say was sometimes there will be molecules which will mimic other molecules. They might be similar in shape, they might be similar in size. So instead of, if we take this example where blue transporter protein had to transport a blue molecule, they are in the same color. But because the red molecule also had similar shape, it failed to recognize and it starts transporting the red molecule. Um, I think, have you guys heard of carbon monoxide poisoning? Yeah, and it is also, as far as I know, more stable than the oxygen binding. Like when hemoglobin binds to carbon monoxide, it is more stable than hemoglobin binding to oxygen. So it is like it will not leave at all. So that is also one of the examples if you are looking at transporter protein defects. And there are nucleic acids. So if you want to target viral genes, for example, reverse transcriptase gene. Everyone heard of reverse transcriptase? Have you guys learned of viral cycles? Life cycle of virus, lytic and lysogenic. Yeah. So if you're going to target those kind of genes, or there might also be some other gene fragments that might induce cancer. For example, I think this is also, for example, if this is the normal cell, you have the nucleus here, and then there is a gene, which is normal gene. If by mistake one more gene fragment, let's say it belongs to humans, but it's mutated or it's abnormal, it just enters the nucleus through bacterial or viral means. It's going to affect and it might induce carcinogenicity. Hope you guys understand. Carcinogenicity is like ability for cancer to occur. Yeah. So in those kind of cases, your target will become gene. So the target can be different. It is not always a protein. And again, you have different actions of the same drug. So I'll just create a new file. If you guys are making notes, uh, do make sure to make notes. So there is something. So we are going to now look at, let me just type in the chat, uh, action of drugs. So drugs can act in different ways. So one is where it is called as agonist. So here what happens is if this is going to be your target, the drug molecule will come and it will bind to the target and it will induce a biological response. Are you guys able to understand what's happening? Is my voice breaking again? Okay, I think, I hope my voice is audible. So this is the example for agonist mechanism, where there is a target And then there is drug. So drug will come and bind to the target, and it will induce a biological response which is usually positive. For example, if I have to give you an example, if this is opioid receptor, have you guys heard of opioid? It's, I think, a kind of a drug. Yeah. So if the target is opioid receptor and you design a drug that is morphine, hope you guys have also heard of morphine. So what it does is it will induce pain relieveness. The person will not feel pain at all. So it will induce a biological response, and that is an example for agonist mechanism. And then there is one more, which is called as partial agonist way, where there will also be a biological response, but it will not be strong. As in the name it says partial, the response is going to be very, very weak. It is also going to activate the receptor or the target, but the response intensity is going to be very weak. And there might also be targets when a drug comes and binds to the target, it will induce the reverse effect. For example, if I design something here, if the drug comes and binds to the target, let's say the original role of the target was to promote glucose synthesis. Instead of promoting, when the drug comes and binds, it will stop producing glucose. So it is called as reverse agonist mechanism of drug action. Are you guys able to understand? Okay. Now coming to the fourth, there is something called as antagonist mechanism of drug action, where what happens is in antagonist mechanism, again you have the target. So let me just name it as T, and then you have the drug. But here the drug is going to block the target. There is not going to be a reverse effect. Instead, nothing will be produced or no action will happen. Are you guys able to understand? Antagonist and inverse agonist are different. So sometimes we might confuse ourselves, but they are different. And then there is something called as competitive inhibitor mechanism. So in that case, what happens is this is the drug and then there is the target. There is a natural molecule, and this natural molecule should bind to the target, but this drug competes with the natural target and it will consume the target's binding space so that there is no space left for natural molecules to bind to the target. Are you guys able to understand? This method of drug action is called as competitive inhibitor mechanism of drug action. And have you guys heard of active site? Okay. So if there is a target here or an enzyme, there is a region called as active site where the molecule should come and bind to induce maximum response or to induce good response. So if I can just write active site here, I'll just write AS. In non-competitive inhibitor mechanism, instead of drug binding here, drug will bind to somewhere else. It will not bind to active site, although it's inhibition, but it is going to be non-competitive. Natural molecules still can come here and bind to the active site. The drug is going to bind to non-active sites. Are you guys able to understand? This is called as non-competitive inhibition. And then there is something called as allosteric modulator. And again, there is positive allosteric modulator and a negative allosteric modulator. Have you guys heard of this term, allosteric? I think we learned this in 12th. Okay, okay, that's okay. So what happens is, this is also one kind of target. For example, I'll just use two different colors. Let whatever I have drawn in black color be the enzyme. Only when molecule comes and binds here on top where there is active site, I'll just write AS, there is going to be biological response, maybe increase in the production of a product. Some targets will have one more site called as allosteric site. So allosteric site, I'm just writing this in blue. It is not that it always is present on the opposite of active site. It can be anywhere, and that site is called as allosteric site. So this is not the active site of the target, but what happens is when a molecule comes and binds here to allosteric site, the effect of the target is going to be uh, affected. For example, in positive allosteric modulation, when a molecule comes and binds here, it is going to increase the production of the product further. There is going to be more product formation than the usual. In case of negative allosteric modulation, if a compound comes and binds here to the allosteric site, the production is going to decrease. Are you guys able to understand what's happening? This is called as allosteric modulator, and there are two types. One is positive and one is negative. And then you have something called as blockers. Blockers are the drugs which will not have any effect, but they will just block the target, either enzymes, or they can block transporter proteins, or they can block uh, ion channels, something like that. That is called as blocker mechanism. And then you have activator mechanism. Some drugs, they can activate receptors. For example, if I take insulin drug, whatever people take for insulin, if this is the cell, you have insulin receptor here on the cell membrane. Insulin drug will not enter the cell. It will come and it will bind to the receptor, and once it binds, the cell signal transduction happens. It is going to sense that insulin has bound to this receptor, and it is now my job to intake more glucose. You have something called as glucose gates. I think in science they call it with a different name. There is a scientific term again. That is going to be more opened so that more number of glucose molecules can enter the cell and can enter the energy synthesis pathway. Are you guys able to understand? This is called as activator mechanism, where the drug molecule will come and it will activate the receptor or a protein or an enzyme that is going to in turn have an effect on the system. And then you also have some drugs called as binders. So let me just type. You have some drugs called as binders. They simply come and bind to the target. They don't have a particular effect. It is not that they're going to particularly increase or decrease uh, a particular, like a production of a molecule, or going to cause any severe effect on the process. They just simply come and bind to the target, and they're called as binders. And then there are few drugs which itself can act as target, which itself can act as substrates. So yeah, so if this, I think this can be a good example. Let's say there is a glucose shortage in my body. If I take the glucose directly as it is, that itself is going to become the substrate. Are you guys able to understand? If I'm suffering from like energy loss or weakness and I take glucose, the glucose molecule will itself act as a drug, and it's also the substrate for the body. And then you have something called as inducer. Inducer. Inducer is going to increase the activity of the target. It usually acts on enzymes. So if I just create a new file again, so if this is a target and I have the inducer come and bind to the target, it is going to increase the activity of the enzyme. So that is going to be the work of inducer drugs. And then you also have down regulators or suppressors, which will decrease the activity or expression of a gene or production of a molecule. Are you guys able to understand whatever I told? I think I covered around 12 to 13 different mechanisms. And it is not that the drug alone acts, there are cofactors, there are other molecules, modulators, etc. They act together. It is not that just one molecule acts on the target and causes the effect. So it doesn't work like that. And if we come back again, so this was all about the target. And then let's just check one pathway, and I'll just tell you how to interpret whatever is present. Select pathways in the drug bank, and then write insulin signaling. And you get this, and it says insulin is responsible for regulation of glucose levels in the body. Everyone got the same result? Click on the first one, which is insulin signaling. And you will be redirected to this website, SMPDB, and you can see this. If you can see, I'll just zoom in. You have got pancreas on the top, beta cells in the pancreas, they produce insulin, and once insulin is secreted, you have got the insulin protein here. Insulin protein is going to come and it is going to interact with the insulin receptor present on the cell membrane. Did you guys understand? And if I just zoom out, you have the cell membrane structure, and you have mitochondria here. You also have nucleus here. And on my left, you have glucose transporter. Are you guys able to see? There is family 2 facilitated glucose transporter member 4. Yeah, so what happens is you have a whole set of reactions mentioned here. I'm not going to cover all of that. So when insulin comes and binds to the receptor, the signal is going to be sensed, and the sensed signal will increase the intake of glucose through this transporter so that more number of glucose molecules will come inside the cell. And once it enters the cell, if you guys remember, phosphorylation happens. It becomes glucose 6 phosphate, if I'm not wrong. So the reason why it becomes glucose 6 phosphate is to prevent it from flowing backwards to the outside of the cell. Are you guys able to understand? Once the glucose comes inside, it should stay here. Hence, it suddenly converts itself to glucose 6 phosphate. And then you have the whole glycolysis, Krebs cycle, electron transport chain to produce energy, and that is going to happen. And again, on the left, you have blood vessels. Hope you guys understood that as well. Blood circulates nutrients to the cells. And on the left, you have description. It says a protein pathway in Homo sapiens, and the title is insulin signaling. You have got a brief description about what's happening in the flowchart that is given, and then you have got the references, attachment of research papers that support this flowchart or that confirm or prove the flowchart. And then if you click on the highlights, you have something called as show in black and white. I would ask you to click on that so that you will be able to understand this better. If you click on show in black and white, the whole flowchart now is present in black and white color. So if you click on D-glucose, only the D-glucose compound present in the whole pathway is going to be highlighted so that it becomes easier for you to track where the compound is and what exactly is happening with the compound. And if you want to choose multiple uh, compounds, you can choose. I have chosen magnesium and glucose, and phosphate and glucose, and you can see there is D-glucose entering the cell, and then there is phosphate here. The phosphate is going to interact with the D-glucose and it forms glucose 6 phosphate. And you have two different categories or two different sections, one where you can just highlight chemical compounds and one where you can highlight proteins. For example, if I just unselect all these compounds, I can just select insulin receptor and insulin. You can see in the whole flow chart, there is just one insulin receptor for a cell, and then only one insulin has been visualized in the flow chart. Are you guys able to understand what's happening? Okay. Now coming to this analyze tab, you have again two different sections, visualize compound data and visualize protein data. And if you can read what is written here, it says red color represents lowest concentration and green color represents highest concentration. So we have to enter the concentration values, relative concentration values without units. So I have no idea what to write. If I just write 16 here, it is in yellow color. So maybe whatever data it has, the 16, whatever I have given as an input, if it compares 16 with the concentration of other molecules present, it is medium, it is neither high, it is nor low. If I just write 20 here, now suddenly the glucose concentration is represented in red color, maybe more the concentration of guanosine diphosphate, it is going to reduce the concentration of D-glucose. So you can check by changing the values of concentration for different molecules, you can check the effect of relative concentration on the system. Are you guys able to understand? Like if you increase the concentration of one molecule, what is going to happen to the concentration of the other molecule? That is something that you can check. If I increase the concentration of guanosine diphosphate, the concentration of glucose is going to decrease. That's why I have got the red color here. As it says, red is equal to lowest concentration. And you can just enter the concentration values without units for all the compounds. Again, you can play with the protein data as well. Similarly, you have to write a number, the concentration without units, and you will get to know the relative effect on the system. Are you guys able to understand? Okay. So if you click on downloads, you have few different options to download this flowchart. That is something that you can try. And in settings, you have different types of visualization. It is just like, um, we have filters in mobile, in Snapchat you have filter, in Instagram you have filter. So it's something like that. So if I select this show keg like font version, I have got nothing. If I select this is the kind of representation of the flowchart. It is slightly changed. And if I select large version, maybe I think it will take time. And then you also have an option called as turn on complex membranes. It did not work for me. Maybe it will take time. You guys can try that as well. Are you guys able to understand whatever is happening within the drug bank? So we have covered drugs, we have covered targets, we have also covered pathways. So the only thing that we are left with is indications, and we will look into this in the next session. It's already 10:34. I think we should not extend the session more. Let me just take a screenshot. Okay, any doubts in today's session? Were you guys able to understand? Okay, okay, that's great. And yeah, so this is all about the drug bank, and it's very interesting to actually like look into the drugs, maybe the common ones that you take when you are ill, and looking into what are the side effects of those drugs and what is the mechanism in which the drug is, affecting your body, something like that. So that is interesting to learn, and you guys can look into that. And obviously, as I told, the open neura, that is also something you can look into.